introduce Natalia Komarova from UC San Diego, doing essentially part 2 of your stem cell contribution. So thanks a lot for this, Natalia.
Thank you. I'm glad to do this. Okay, I will go over some of the slides, right, and remind you what I did before. So can you see this well?
Yes.
Okay, so we're talking about adult stem cells, that everybody has them, and we are particularly interested in tissue, on epithelial tissues. For example, skin, also colon, and some other ones that I'll show you. Stem cells are here. This is the upper layer. Stem cells divide, and in the process of division, they somehow change, and such that one or both offspring cells become different, travel upward, and continue changing until they're here. This is the upper layer of the skin, and these ones perform their function. Okay, so many types of stem cell lineages have been identified. This is blood, and we will return to that in greater detail today. So these are the differentiated cells. This is a stem cell, okay, and they communicate with each other. So I think this whole talk and this whole topic is centered around communication of cells, and how the communication can be broken by a mutant. So this is colonic crypts like this, and stem cell sits here at the bottom of each. 10 to the 7 of these folds of colonic tissue are in a human, and there are 10 to the 4 cells in one of those. So it's very, very small. Neural, olfactory has to do with the sense of smell and so on. We showed that control or communication among cells is necessary because when the stem cell makes a decision on whether to differentiate or just self-renew, it in the absence of communication, we have a symmetric random walk or a drunkard's walk, which is not a good idea if your variable is the number of stem cells. You don't want it to disappear or to go to infinity, basically. So you have to establish some control or communication among cells such that the stem cells are in check. They need information from their environment to know when to stop dividing, right? And many control networks have been envisaged by theoreticians and experimental biologists. Most of them are still not known. So we figured out that there is a kind of a general theory in the absence of detailed knowledge on how these things communicate with each other, how they control each other. We can still say something about the structure and organization of stem cell lineages. And this is done by means of saying that all the rates are functions of the populations. So the population of stem cells we call X, population of differentiated cells we call Y. Their different decisions are denoted by different letters. In this simple system, there are only 2 types of cells and only 3 types of processes: division, probability of differentiation, and death. Basically, once I decide to divide, I can either divide into 2 of the same type or 2 of the next type. So this is a very kind of common paradigm in human epithelial tissues. And also the differentiated cell may decide to die. So these rates depend on the populations because they're controlled by the other cells that are there. And we talk about partial derivatives. So how is the division rate defined by the number of stem cells? It's a function of X. So how does it depend on it? If there are more stem cells in the system, will they decide to divide more or less? Well, that is determined by the sign of the partial derivative. If this rate dl dx, the derivative is positive, it means that the more of stem cells they are, the more they divide. For this one, it's an example. How does the division rate depend on the number of differentiated cells? In this picture, dl dy is negative. The more of them are, the fewer divisions of the stem cells we are going to see, because they signal and they say stop. Okay, so this is a way to represent control or cell communication by means of partial derivatives. And so these are functions, right? But we evaluate these functions at the equilibrium. So it's not a function, it's a number. Okay, and the last thing that I wanted to kind of review with you is how to construct. Oh, actually, we don't have to do it. I'll show you again. So for these very simple systems with this type of control, with these processes, we were able to. Sorry. Okay, we were able to say that there are the simplest ones with these arrangements of the loops. And in the presence of 3 compartments, there are more, but using clues from experiments, we can narrow this down, right, to get to some reasonable number of these cells. So this was this work where we ended up with only 2 types of networks that we think could be operating in colonic tissues. And the variance should be minimized. So when the number of cells oscillates or fluctuates around its equilibrium, it's probably good from the point of view of the organism to minimize these oscillations. And based on that, we were able to kind of narrow down networks that operate in airway epithelium in mice. Okay. So now we're talking about breaking networks. That's where I stopped last time. So I want to go over the system a little bit. This system is different from what I presented before in 2 ways. And it's the same in maybe 1 way. So it only contains 2 types of cells: stem cells and differentiated cells. In this paper, it was also generalized to 3 cell types. So that's the same. What are the differences? More processes are considered. So here we have 7 different processes. And also, we assume that all the rates are per cell, not total. That's why we have X, the number of stem cells that appears in front of all the rates that are associated with the actions of the stem cells, and Y, the number of differentiated cells, appears in front of its own rates. So let me talk a little bit about the system. By the way, can you see the right-hand side of these equations? Because for me, it's not very good. Let me just...
Yeah, we see everything.
Can you see the right-hand side of these equations?
Yes,
Yeah.
we see
Oh, very good. Thank you, because they're blocked for me with the gallery. Okay, so how does the number of stem cells change? It changes because there are processes that happen. Let's look at R. R is the divisions of stem cells that are differentiation divisions. When a stem cell divides and differentiates, the number of stem cells decreases by 1, because a cell splits into 2, but these 2 are different color, right? Not different, they have different properties. X splits into 2 Ys. So the number of X's decreases by 1, but the number of Ys increases by 2. So we have X decreases at the rate R, but at the same Y increases by the rate R times 2, because 2 of these cells come here. So from here, 1 is subtracted, and here 2 appear. Okay, so that's differentiation divisions. Let me also do self-renewal divisions. This is the second type of divisions that stem cells undergo. Now, if an X divides, it creates 2X. So what happens here with this process is the number of X's increases by 1 and the number of Y's doesn't change. So we have this term here, stem cells. Should be minus here. It's a positive term that increases the number of stem cells. Okay, and it doesn't appear here. Yes. What else? Asymmetric divisions. So when a stem cell divides asymmetrically, what happens? The number of stem cells doesn't change because one of the daughters continues to be a stem cell, and the number of differentiated cells increases by 1. So the number of X's doesn't change, the number of Y's increases by 1. So the rate A for asymmetric divisions appears here once. It's proportional to the number of X, the stem cells dividing, and it increases the number of differentiated cells. What else do we have here? De-differentiation of differentiated cells. So one of these Y cells can go and become X. Okay, so this term called B takes away from Y and comes back to X. So from here to here, you can see that in the second equation, it appears with a minus sign and it goes back here. For completeness, we also included the divisions of differentiated cells. Okay, we assume that they can divide. That's this term. It comes with a plus sign, and it increases the number of differentiated cells by 1. And finally, 2 death rates. The death rate of stem cells and death rate of differentiated cells. So this is death rate of stem cells takes away 1 cell from X, and death rate of differentiated cells takes away 1 cell from the Y's.
So Natalia, there's a minus sign as well, right?
This
sign should be
so
plus.
these are, yes, exactly. So I think this should be minus and this should be plus. Thank you. I should correct it because I've had it forever. Hopefully it's correct in the paper. Okay, so this is how these terms enter into the equations. And we can simplify a little bit by combining them because some of them appear in exactly the same way. Let me see. So we combined the self-renewal. We combined SCS and DCSC like this. And we combined the death of differentiated cells in their divisions with a minus sign. So if we do that, we have fewer terms in the equations, okay? And so this is the system we'll be working with. Let me put this here, okay? Now we have to remember that all of these 5 remaining rates are functions of X and Y. And what can we do with such a system, right? So we are first going to assume, sorry, what is going on here? We are going to assume that this system has an equilibrium. Don't try to solve the system as it's written, because as it's written, we don't know what R is. We don't know what S is. And it looks like it's a linear system, right? That X multiplies that, Y multiplies that. This is misleading a little bit because these are functions of X and Y. It's a nonlinear system. And unless we know exactly what the functional form is, like what is R as a function of X and Y, we cannot tell what the equilibrium are. We're going to assume that there is an equilibrium solution of this system of ODEs. But once we assume that it's there, we can investigate its stability, you see? How do we even do that, right? So in order to investigate stability of an equilibrium, we have to write down the Jacobian of this. Hopefully you are somewhat familiar with this theory of linear stability analysis. We take the equilibrium, perturb it, we write down the Jacobian and calculate its eigenvalues. the sign of the real part of the eigenvalues tells us whether the equilibrium is stable or unstable. So our goal, if we have an organism that we're trying to build, is that the equilibrium is stable, because if, you know, we want to have homeostasis here. And the next step here will be to break it, right? So now we want to have something that corresponds to a healthy state. So the question that we ask is, what is the minimum number of control loops that we could have in the system such that we could guarantee that the equilibrium is stable? So what's a control loop? So this is an example of a control loop that works. This says that the rate R, which is differentiation of stem cells, is positively controlled by X. This means that here, this function R, and it also appears in the second equation, this function is a function of X. And moreover, dr dx, the derivative, is positive. The more x's there are, the more they try to differentiate. And what else this says is that all the rest of these parameters, S, B, and D, they are constants. So, right, so the presence of only one loop in the system is enough to guarantee the stability of the equilibrium as long as its derivative with respect to x is positive. So this here is a notation for a partial derivative taken at the equilibrium, dr dx. And if it's positive, then this loop guarantees that the equilibrium is stable. So you can do this as an exercise, assuming that a, d, b are constants, r is a function of x such that the derivative is positive, calculate the Jacobian and prove that both eigenvalues have a negative real part. Okay, so if b is 0, there are 4 such cases, 4 types of loops that will guarantee stability. For example, this one says that self-renewal of x is negatively controlled by y. So in this system, only s here is a function of its variables, and in this case, it's a function of y, and ds dy is negative. If we add the possibility of de-differentiation, we have 6 more control loops. These are minimal control loops. Each of them only has one functional dependence of one of these rates on one variable, basically. So these are the minimum control loops that are compatible with stability of the equilibrium. Any questions about this? All good? Okay. So now, what kind of mutations can break this system? So we're going to consider 2 types of mutations. Mutations that don't participate in signaling and mutations that do not respond to signaling. So here, mutations that don't produce a signal. So if a rate, say R, should depend on X, a mutant can arise that doesn't contribute to the strength of that signal on X. And here, mutants that don't respond. So if your rate of divisions should be a function of Y, you don't care. And you're just going to keep dividing, or you care less, right? It's a function that depends on that to a lesser extent. So we can show that mutants that don't participate in signaling are not dangerous mutants. You can prove that they are neutrally stable. When we perturb the corresponding equilibrium, most eigenvalues are negative and one is 0. One or more is 0. However, these are the dangerous ones. These are the ones that don't listen. Okay. So how can we show this, formulate this mathematically? So this is the same system as I had before, except now I say X1 and Y1. These are normal stem cells, and these are normal differentiated cells, right? Or we can say normal or wild type And these are mutants when I put the subscript 2. So, and you can see X and Y that appear here. This is the sum of normal and mutant stem cells. And this is the sum of normal and mutant differentiated cells. So this rate of self-renewal here, I'm sorry, the rate of differentiations here is a function of all the x's and all the y's, right? And when we look at the normal cells, they function according to these equations that we already looked at. Now, mutants have something different about them. You see that it's exactly the same equation, but when I say the differentiation rate of these cells, how does it depend on stem cells and differentiated cells? There's a factor here. Think of this as a number. And let's suppose that this is 0, like this is 0 and this is 0. What does it say? It means that this for the mutant cells, the rate of differentiation is a constant. It doesn't depend on what other cells are telling them to do, right? So in the most extreme case, all these constants are 0, and mutant cells just don't care. They don't respond. Or you can say that, okay, this is a number that's between 0 and 1. They care somewhat, but less so than these. So it's a flexible system that allows us to go between not caring at all or caring almost all the way, right? But these dangerous mutants don't respond as well as the normal cells to controls. This is the most general system. In all of these, only one of these functions is really a variable, a function of only one variable. So in a specific case, like all these are constants except r, and r is a function of x here, r is a function of x, and here r is a function of gamma x. And if gamma is 0, it's just a constant, right? So this is how we can formulate it. And stability analysis here involves 4 variables. So when we only had 2, that was easy because everybody can calculate eigenvalues of a 2x2 system. 4x4, not so easy, but still possible. The first question we ask is there a better or worse system of controls? Are some of them more difficult to break? And when I say break, this is what I mean. When I introduce some mutants, so if x2 is equal to 0 and y2 is equal to 0, everything is great. We just have a well-functioning, healthy control system. Let's introduce some mutants, and the question is, are they going to grow? So, and if they start growing from small numbers and kind of overtake the system, that means that the healthy controls are broken, and these selfish cells that don't want to respond to control are growing. Okay, so are any of these loops better than others? Okay, so to do this, we're breaking the single existing control for each of them and looking at the dynamics of the mutants. Are they going to grow or are they going to like stop? And the answer is no. None of these is any better than others. They're all created equal. Any loop is equally vulnerable to an invasion of mutants that don't respond to control. Okay, so for example, here, this is a simulation where we take this system and assume, okay, r is a function, like this one, for example, r is a function of x such that it grows with x, so stick in some growing function of x and simulate it, right? The rest of the parameters are constants. We start from an equilibrium, so these are normal wild type stem cells, and these are normal differentiated cells. So this is your equilibrium, and we put a little bit of a mutant here. And after a while, you see that the mutants come up These are mutant stem cells, these are mutant differentiated cells, and the original cells die out. So here mutant took over. And there are several, for each of those control systems, you can have a similar picture. Mutants always take over. That's a fact of life. I want to attract your attention to the generality of this result. These simulations are not general because in order to draw a picture, you have to assume the functional form of these dependencies. But the theoretical results are completely general. It doesn't matter what R is, as long as this is a growing function of X, and there's which guarantees the existence of a stable equilibrium. You put a mutant that refuses to respond to this, the mutant will grow. So very general result for all of them. So how can we protect the system from the invasion of these mutants? How can we do better? So one solution that nature often uses in different contexts is redundancy. Before we looked at the minimal loops. Having these loops is expensive, so you can't just control everything, because control means that you have to send the signal and somebody else has to read the signal. All of these are adaptations, and if you can do that, it means that you can't do something else, right? Where we all have finite resources. So we would like to get away with the smallest number of these loops. But maybe the smallest number is not a good idea because an invader always comes and takes over. It's easy for it to break a single, protection barrier. So let's try to combine two. So we can consider all the combinations of two loops like this. It's that loop and this loop. And so we can classify them in the following way. So we're going to look at the original number of loops that are compatible with the stable equilibrium of normal cells. There are 10 of them, so there are 10 of these loops. And each of them can be combined with any other loop, right? So for example, if we take this one, it can be combined with this loop, not with itself, so we're going to ignore this. It can be combined with this, that. So these here are loops that are eliminated by mutation, and these are the loops that stay intact, right? So, and there are all these different combinations. Gray rectangles means that it's self, so it's not a combination of two different loops. So we ignore the gray. And there's some color code here that tells us what happens when a mutant appears that eliminates one of the existing loops. So these are the equations that we are solving. And let me explain the color code. So let's first look at the arrows. So some of these cells contain arrows. Arrow is the change in the fraction of differentiated cells per stem cells. So I don't like anymore the way it's arranged. I would rather, if I did it again, I would say X over Y because it's more informative. So basically, we want to track enrichment of stem cells. So if the number of stem cells in the system increases, then this quantity will decrease. So every time we see a downward arrow, that means that a mutant, the existence of a mutant, leads to the enrichment of stem cells. And normally this is bad, right? Because stem cells are great. We should be grateful to stem cells for maintaining our organs. But too many stem cells is not good because they are harboring the infinite division potential. And infinite division potential is not good if something goes slightly wrong, right? Also, for many cancer treatments, it's the stem cells that are the most difficult to target. So the more of them they are, the worse. So every time we see an arrow that goes down, this means that in this particular combination of two loops, the existence of a mutant will lead to the enrichment of stem cells. Okay, so now colors. Red means that a mutant will give rise to both populations increasing. Yellow means that stem cells will increase. Blue means that differentiated cells will increase. And green means that only one of the populations will increase and the other may decrease. And let me show you. So, the equal sign means that the fraction doesn't change. And question mark means that it could go either way, right? It's not that we don't know, but it could go either way depending on the particular functional form of these controls. So let me show you some of the green ones. Greens are good, right? So let's take a look at this one. We start with an equilibrium of normal cells. These are stem cells and differentiated cells. And then we introduce a mutant that breaks one of these two loops. I don't remember which one. So we start with these two loops. And so there is some initial adjustment. And you can see that even the mutant will drive the normal cells away, okay? Now there are mutant cells. But you can see that things don't go to infinity, and that's good. And also, you can see that even though the number of differentiated cells increase, the number of stem cells decreases. You can't see it very well here, but it's actually less. So this combination of loops is not easy to break in a bad way, right? It's still kind of there. And here's another one. This is a different combination. So here, the rate of self-renewal here, it's negatively controlled by differentiated cells. Here it's negatively controlled by stem cells. This is also a good combination. And as a result of a mutation, you can see that the number of differentiated cells increases, but the number of stem cells decreases. You can clearly see it here. So not all combinations of loops are created equal. So there are redundant control systems that are less vulnerable to mutations. So that was interesting to see. And here is a slide that is not from my work. It's very famous conceptual framework of Yuri Alon, who studied network motifs. So he looked at different biological systems, for example, gene regulatory networks, neural circuits, protein interaction networks. So these are other parts of biology where there are networks involved, right? Where some entities are connected by arrows that mean something, mean some signaling, mean some communication, right? So he looked at all of them and he identified that there are motifs in these networks that occur again and again and again. So theoretically speaking, there are many, many networks that are possible, but only some of them keep occurring in nature and not others. And this is kind of what we see as well. There are many theoretically possible networks in the part of biology that we looked at, the communication among stem cells and their lineages. In the colon cancer work that I showed you before, we were able to narrow that down to only a couple. So we would like to see more of this type of work done to see if we can figure out what are the most commonly occurring networks of communication that exist in the world of stem cell lineages. Maybe a theory like this can be possible to develop as well. So when you study these things, it's nice to make a connection to wider areas of biology and see some common patterns, perhaps. Okay.
Natalia, may I ask you a question?
Please.
Before we move forward? I just because you were focusing on the fraction like x divided by y or the other way around, doesn't matter. But it can be constant, but then you can have x and y growing, let's say,
Absolutely.
and then you can end up with a polyp just because everything is growing. So I don't know if it's I don't know if just looking at the fraction being
Oh,
constant.
so the fraction is only part of this information, right? So we actually looked at everything, but I wanted to concisely indicate the behavior of each of these controls. So the arrow or the equal sign or the question mark refers to the fraction. Let me go back here. Sorry. Where is this? Here. So the arrow or the equal sign or the question mark refers to the fraction. But the color talks about the absolute values, right? So red means that they're both increasing. This is exactly the situation that you just described. For example, this one, the fraction remains the same, so that we have equal sign, but both populations are growing. So that's exactly what you described. But for me, the interesting part here was the existence of green ones. That means that like at least one of the populations decreased. So that was cool. So there, does that answer your question?
Yes, thank you very much. And I
Very
think there
good.
was another question in the chat
Oh,
that we saw.
okay, can you please voice it? I don't know how to read chat from here.
Um, well, yeah, the person who wrote the question, perhaps you can just say it.
Yeah,
I won't
thank
remember
you.
the question.
I was just wondering when you like presented these gammas that you multiply x by. So you were discussing the case that they're between 0 and 1.
Yes.
And like at that moment, I was just wondering, like, what if they were greater than 1? In that case, the mutants will be carrying more than the wild type, but I guess maybe
So
they will
great
die.
question.
But I don't know if there's another possibility other than dying.
So that's a great question. So mathematically, we can look at. So this is gamma r, a number that multiplies the control of differentiation rate by x the stem cells, right? So if gamma r is equal to 1, that means that it's the same control loop as in the normal cell. If gamma is 0, it means that it doesn't respond at all. If gamma is the number between 0 and 1, the one that I described, it means that it responds but less, right? What happens if gamma is a number that's greater than 1? It means that it responds more. So if x's are telling me don't divide, right? So as x increases, I divide less and less. But the mutant that has a gamma even bigger is going to panic and say, oh my God, I can't divide anymore. And this mutant is going to stop dividing even before normal cells. So these are going to die out, actually. Such mutants, maybe some work can be done to prove it rigorously, but my intuition is that such mutants are not going to do well in terms of survival. As opposed to being selfish mutants, these are selfless mutants. They're the ones who are so responsible, they do more than their fair share, and they will not have an advantage. They'll have a disadvantage in this system. They will be selected against, and we will never see them. So if you start with an equilibrium characterized by normal cells and introduce a little bit of those mutants, this perturbation will die out. Does that answer your question?
Yeah, thank you.
Thank you. Any more questions? Okay. So, where were we? Here. Okay, so I want to now show you a specific problem that has to do with blood. Okay, so this is a diagram that I already showed you before. Stem cells differentiate into other types, and here we have a split between two important lineages. These are immune cells, and these are just cells that comprise the blood, like platelets, granulocytes, red blood cells, and so on. Okay, so we are going to focus on these first 5 types. All of them are sitting in the bone marrow. These are in the blood. Okay, so we're going to look at the root of the cells that make blood. This is called long-term hematopoietic stem cells, so long-term stem cells. These are called short-term hematopoietic stem cells. These are MPPs, I forgot what that stands for. And these are the 2 lineages. Okay, so I take that bit and put it horizontally. Long-term stem cells, short-term stem cells, MPPs, and then 2 lineages. Okay, so I drew this differently. And we are going to look at this specific system and use data to parameterize it. So this will be a very specific application of these theories. Okay, and we're going to talk about 2 types of divisions again: self-renewals, when a cell divides and creates the same type of cells, and differentiation divisions, when you create the cells that are different, so they go here. So self-renewals return to the same compartment, differentiations take the offspring of the cell and place them into the next compartment. So this is the system of equations that describes all these processes. And it's very similar to the system that I showed you before, except there are fewer processes. We don't have de-differentiation. We don't have asymmetric divisions, although mathematically they can be included without loss of generality. We don't have death in these compartments. Death definitely occurs, but further down the stream. This D here is not death. It's like these go on. Remember, they go on to these other lineages. We just say, okay, they're removed from here. Whatever happens to them, we don't consider here. Okay. And so I derived these systems, exactly these systems last time. So this here, when a stem cell, X0, this root stem cell divides and differentiates, it divides with rate R0 and differentiates with probability 1 minus P0. So R0 times 1 minus P0. And the divisions are proportional to how many cells they are. And they create 2 of these cells. So this term comes from here with a factor of 2. And it's subtracted from here. 1 minus P0 is subtracted from here. And this term is self-renewal of these minus differentiation of these. And differentiation goes here and so on. And these 2, just there is a removal rate. Okay, so theoretical system, all the P's and all the R's can be functions of every type of cell. Okay, so now can we say something about these parameters and what they actually are? So let's suppose that we are at the equilibrium, healthy equilibrium of a mouse. At the equilibrium, we can ignore the fact that this is a function, could be potentially a function of all the x's, right? And we say, okay, it has a certain value at the equilibrium. What is that value? So this is what we were able to do. We used the data that appeared in this paper, experimental paper, and this is a very ingenious method of measuring things in a live mouse. So they put some substance into the cells in the most upstream compartment, long-term hematopoietic stem cells. And this substance is what they call a neutral label. It means that a fraction of these cells were marked, but otherwise they had completely the same properties as the unmarked cells. So neutral means that they don't differ from the other cells by their division and death rates or renewal rate. All the kinetic parameters are the same. They're just there and they're colored and we can track them. So what happens if we put such a thing in this upstream compartment? Well, but some of these cell divisions of the purple cells will bring them back here, but some will take them here. So we're going to have the label leaking here, and you can detect it in this compartment, and there'll be more of it here with time, and these will differentiate eventually here. So we'll be able to observe some of these cells here, the progeny of these labeled cells, and so on. And after a while, we are going to reach a steady state. Okay, what is the steady state going to be? You can see, like, just from intuition, the fraction of labeled cells in each of the compartments will be equal to the fraction of labeled cells in the original compartment. And as time goes by, the fraction of the labeled cells was measured among all the cell types. So we have 4 different lines here. The 4 lines correspond to these 4 compartments, not the original compartment. They had a line for that too. It's more or less constant. It's, the label is just sitting there, and it's spreading through. And after a while, it reaches a saturation, an equilibrium. Okay, so for example, this here, the blue line, is the fraction of the label in short-term cells in this compartment. You can see it's higher originally than all the other ones because the label spills here first. Okay, and these follow. And of course, you can see that the data are noisy, but that's normal. This is biology. We can see the general trend of these things increasing, of the short-term stem cells leading, so it all makes sense, and eventually they saturate. Okay, so by using these data, we can take our equations, these equations, and rewrite them for the fraction. So it's really easy. You just say, okay, these are normal cells and they're labeled cells. They satisfy the same equations. Let's form a fraction of labeled to unlabeled, or labeled to the total, and write down the ODEs for the fractions. And these ODEs are linear, very simple. So for example, in the most upstream compartment, the change is 0. And then
Excuse me, Natasha.
Yes,
Is
hi.
it possible to interrupt you to ask a question? Where do I see the hierarchy? Like, where is the hierarchical structure hidden here? Can you remind it, please? Because you referred to hierarchy in the previous slide.
Yes, so the hierarchy is reflected in the subscript in the equations. So 0 corresponds to the most upstream compartment, 1 to the next one, 2 to this one, and we have 3, 2 of them, and they're labeled as 1 and 2 because it stops being linear and it branches or splits into 2. So we only have 4 levels, 0, 1, 2, and 3, and there are 2 types in the 3rd level. So this is what we mean by hierarchy. It only goes one way. We don't have de-differentiation, so all the arrows come here. So this equation spills over here, this equation spills over here, and there's nothing going back. Does that answer?
Yeah, it makes some sense. And for the branching, so you have branching at different levels. Of course, you have branching at different levels of hierarchy. Yeah, I can see it.
No, there's only one point of branching. This is the here. Oh, actually, there is more branching going on later on.
Yeah. That's what.
But we stop here because we're looking at the bone marrow. These are the types, and eventually they leave bone marrow, and these are in the blood. Okay, so we only have one branching point in this system, and then we'll cut it. There are no data for the next compartment. So, okay. So when we rewrite the system in terms of the fractions of the neutral label in each compartment, we get a linear system. And you can see that there are not so many parameters in the system. There's one here, one here, one here, and one here. So with this data, we are able to identify these parameters. They're called taus in this, but they're really just combinations of the parameters that appeared in my original ODEs. And so using this data, you can calculate what they are. So this is us fitting the data. So these 4 graphs contain the same information, and in this one graph only split into the blue type, the yellow type, and so on, right? And the red lines are the fits. So we can calculate all of the parameters. And this turns out to be important, actually. You can say, oh, who cares about the specific numbers, right? But in fact, you will see that there are some interesting biological conclusions that come from that. So let's just say for now that we know what the parameters are, and these are the parameters at the equilibrium. And now we can make the next step and introduce mutants in the system. So this is the end of the experimentally validated part. We have the system with the parameters. We're very happy about it. It doesn't often happen. But now we're going to introduce mutants. And of course, in the original experiments, there were no mutants. So how do we introduce the mutants? This is the original system with X. And now we make a copy of that, call them Y, and these are the mutants. Now, all the rates are now functions of X plus Y, the total number of cells. And the equations for mutants have the same structure as the equations for wild types, but they may have different parameters. So what are the parameters here? There are really 2 types of parameters: the division rate and the probability of self-renewal, P, right? And so I didn't write it here, but in the most general scenario, the mutants will have a different division rate. All of these R's will be different, so we can call them R M mutant, right? I didn't do it here. And the probability of self-renewal is different, M. It has the mark M. And if we want the mutants to be advantageous, we can say that their probability of self-renewal is bigger. So if S is positive, they're more likely to self-renew. But I'm getting ahead of myself. We can just keep this general, okay? So now, what is the fate of a mutant in the different compartments? So let's think about it. Let's suppose I put a mutant that is somewhat advantageous in the very first compartment. What is going to happen? This mutant, by being advantageous, is going to kind of take over this compartment. It's advantageous. It's going to win, right? Because of the force of selection. And once it's won here, it's going to spread throughout the system and everybody will be mutant. Okay, an advantageous mutant that we place in the most most upstream compartment will win over. Now, what happens if we put a mutant here? And that's less clear, right? Or here. So these are the questions that we are going to try and answer. So this is, I repeated the system. Now it has 10 equations. And we are going to perform, to answer the questions that we asked by performing a linear stability analysis. And again, I hope you are somewhat familiar with it. I already mentioned linear stability analysis in the context of my previous slides. So here it's done for this system. So we assume that there is a mutant-free equilibrium. And in fact, we don't have to assume it. We know it's there. So we are going to write our variables in terms of a vector. The first 4 places are for normal. These are for the mutants. And the mutant-free solution has normal cells and no mutants. So this is the type of an equilibrium we are going to perturb. We are going to write down the Jacobian, which is an 8x8 matrix, calculate the eigenvalues and eigenvectors of that matrix, call them lambdas. And we find that if we do that, stick this here, and the existence of zeros helps, right? It makes it a little bit easier. And we have two separate groups of eigenvalues. So these eigenvalues have eigenvectors of this shape. So the eigenvectors have an entry corresponding to normal cells, 1, 2, 3, 4, and no entries for the mutant cells. So this means that these 4 eigenvalues are responsible for the internal stability of a normal equilibrium. In the absence of any mutants, let's take that equilibrium, this one, and perturb it. Okay, maybe we'll like remove a few stem cells or add a few differentiated cells. Will it return back to its normal state? And this is not an interesting question because we know that it will return to that because the hematopoietic system is there. We have a healthy equilibrium. It's robust, right? So these are eigenvalues that kind of take care of the existence of homeostasis in the absence of mutants. And this is of no interest to us. All of these are have a negative real part. But there is a second group of eigenvalues, which is more interesting, and it has to do with mutants. So this eigenvalue number 3 is always negative because of this. Oh, by the way, I forgot to say that I'm making a simplification here. You see, there are really 5 equations here, 10 in total, and I'm talking about 8 by 8. Why is that? It's because I took these 2 that correspond to the last thing that branched, and I combine them together because I don't care about this group. These are kind of the cells that are the most differentiated in my system. So I take the sum of x3 1 and x3 2. So that's why I only have one x3. So I have 4 equations here and 4 equations here. That's why I have an 8 by 8 matrix, not 10 by 10. Okay. So D3 here is a sum of these. Okay, so and the other eigenvalues in this group may or may not be negative, right? But they have this beautiful shape. So it's 2 times the probability of self-renewal at the equilibrium minus 1. Okay, so whether or not this is greater than 0.5 will tell me whether this mutant will arise. If this equilibrium is unstable, biologically speaking, this means that this mutant has the possibility of winning over the system, expanding from low numbers, and eventually possibly taking over the whole system, maybe. If all the eigenvalues are negative, it means that this mutant will die out. Okay. So for stability, so the mutant has no chance, right? For stability, we need this to be negative. So the probability of self-renewal has to be less than 1/2. And if we write this like we did before, 1 plus s times the normal. Oh, wait. So before I go on, let's see how stability depends on the division rates of mutants. So these are mutant division rates. And you can see that I can do nothing with this number to change stability. It's just a factor that multiplies it, unless I make it negative, which I won't. It doesn't make sense. The division rates of mutants have no role in their stability. So that's why I even skip the notation m here, because it doesn't matter what the rate of division is for mutants. It's very counterintuitive, actually. Having a faster division rate usually is associated with advantage, right? But not in this hierarchical system. Here, a more important role than the division rate is played by the probability of self-renewal. This is what defines the sign of the eigenvalues. Okay, so let's focus on this one. I suppose that for advantage, I have to multiply
Natalia,
Yes,
I'm sorry. It's just that they're kicking us out from this room, so we will join online, okay, in a second.
of course. Should I take a small break so you can relocate?
Well, I don't know. It's just gonna take us a minute. I think you can continue.
Okay, thank you for letting me know. Okay, if we rewrite the probability of self-renewal for mutants as 1 plus S selection coefficient times the normal, this inequality becomes very simple. It's a simple equation for S. I'm sorry, simple inequality for S. And this is the measured equilibrium probability of self-renewal that I have in my table, right? We estimated it from the experimental data. So now, let's talk about introducing the mutant in different compartments. So this PI here is the probability of self-renewal of normal cells. P naught is the probability of self-renewal of stem cells, this one. And it's exactly at equilibrium, it has to be equal to 1/2. You can see it from here. If you solve this equals 0 at equilibrium, you see that 2 P naught is equal to 1, P naught is equal to 1/2. So stick it here, 1/2, and you can see that the left-hand side is always bigger than the right-hand side. This is 1/2. So any advantage will destabilize the system if it starts here. Okay. So let me remind you how to proceed with this analysis. Once we have all the eigenvalues and all the eigenvectors, the solution of the linearized system can be written like this, where these are the eigenvectors. These here in the exponent are the eigenvalues times t, and these are some constants. This is your solution of a linear system. And the initial condition for the mutant perturbation is 0, 0, 0, 0, and the wherever I put the mutant. So this is how much mutant I initially put in the first compartment. This is how much of the mutant I put in the second compartment, and so on. So this is where it becomes really important where I put the mutant. So if I want to place the mutant in the very first compartment, I take this positive, and these are 0. So this is the initial condition. If I place the mutant here, 0, 0, 0, 0 for wild type, 1 mutant here, and nothing here. Then I can solve for these numbers, and you can show that this solution will grow if S is greater than 0, any S. But now if I put it here, the first alpha will be 0. The alpha that corresponds to this compartment. And I have a less trivial condition for S for it to enable the growth. So this is 1 plus S, P1 should be greater than 1/2. And that means that S, and this is a measured value that's less than 1/2, S has to be greater than 0.058. And if I do it here, S has to be even greater. So we have something that is very unusual. We discovered the existence of invasion barriers for the mutant. If you put the mutant here, there is no barrier. But if you put it here originally, the barrier is 5.8%. The mutant has to be more than 5% fitter than the wild type. And actually, in evolutionary biology, a 5% fitness advantage is huge. Over the years, in the evolutionary timescales, fitness advantage of 0.01% is something that is very significant and will grow over the years, right? So having such a high barrier is a very significant restriction on what types of mutants can invade. And the barrier here is even huger, okay? And so the particular numbers come from our estimated parameter values. So that's why I said it was important to know the actual numbers. Okay, so what is the reason for the existence of these barriers? It's actually very easy to understand. If I put an advantageous mutant here, it competes with the host, it wins, and it spreads. Okay, no barrier. If I put an advantageous mutant here, it competes with the host, but it has kind of a losing position because the host has help. The original cells that were here always receive reinforcement from the upstream compartment. So as a mutant, I can be stronger. I self-renew more, but they always get new normal cells coming in. So I have to compete not on an equal basis. Okay, and the intensity at which they come in defines the amount of barrier that I have to overcome to win over this compartment. And here it turns out that the barrier is even higher. So this is the graphical representation of this idea. No barrier here, a relatively low barrier here, and a very high barrier here for the mutants. Here's a simulation. Let's suppose that we have an equilibrium. So these solid lines correspond to the number of cells in each of the compartments. So this is long-term stem cell, short-term stem cell, MPP, and these other most differentiated cells. So everything is great at the beginning. And we are going to put a mutant here that has a very small advantage of 2%. And the mutants in different compartments are shown in dashed lines. So we put a mutant here, and look, it spreads to the other compartments, and they all grow, and eventually they displace the host. Okay, so a very small advantage of 2%, I put it here, it wins over after a while. Now I take the same mutant and put it here. And this is the simulation. I put it here and it dies out. Why? Because its advantage is not enough to overcome this barrier. I need at least 5.8 or a little bit more advantage for it to succeed. The third simulation corresponds to me putting a mutant here, but this mutant has a 25% advantage. So it's more than the barrier, it wins, okay, and establishes its own equilibrium. So if I took a 6% advantage and put it here, it would not do well, it would go away. So the next slide shows one of these trade-offs that nature faces when designing tissue. We're not talking about real design, we're talking about evolutionary optimization, right? So it has to optimize over different things, and sometimes it needs to compromise. So let's take the self-renewal probability, for example, in this compartment. This self-renewal probability is an important parameter that is responsible for the size of this compartment. If they don't self-renew very much, this will be a small compartment. Not self-renewing means that every time they divide, they go here, right? So the less the self-renewal probability, the tinier this compartment. So as a function of self-renewal probability, the size of this compartment increases. And one of the reasons we even have this hierarchical structure is amplification. We only have a few stem cells. We want to have more of these and even more of these and even more. We have to create all of the blood, right? So amplification is an important task. So I kind of want to have self-renewal probability closer to here. On the other hand, the self-renewal probability is responsible for creating the invasion barrier. And the invasion barrier, as a function of self-renewal probability, looks like this. It plunges. If the self-renewal probability is very close to 1/2, there is no invasion barrier. Look at that. So I don't want to be here. because I want to protect against mutations. So on the one hand, I want to have a larger compartment size, so I'm pushed here. On the other hand, too close here means I'm vulnerable against mutants. So nature solved this compromise by setting this P, the self-renewal probability, at a certain value. And these were the values that we estimated from the experiments. Okay. So here are some simulations of how this protection works. Let's put a 2% as a mutant that has a 2% advantage into the most upstream stem cell compartment. I'm sorry, not most, the second one. The one that requires 5.8% barrier, right? So not surprisingly, this thing dies out. Okay. Now let's put let's include the effect of aging in the system. So what is aging? It has been noted that as an animal or the human ages, the input from the upstream compartments decreases. So you remember that arrow, unfortunately I didn't put it here, the arrow that goes from the most upstream compartment to the next one, it carries cells from the most stem-like state to the next one. It replenishes the cells in the next compartment. That source kind of dries out, not completely, but it becomes less. So here we keep it constant, and here we decrease the influx from long-term hematopoietic stem cells. In the equations, this term was R0, X0. Let's suppose it decreases throughout the lifetime of a mouse. And look at that, the same mutant will now rise. Because the barrier gets lower and lower. The barrier is defined by that influx. So this may be what is happening, because the question is, in the presence of the barriers, somehow mutants still arise, and still organisms get cancer, right? So the question is, how does that work? It's always an arms race, right? Somehow the evolution is trying to prevent cancer, and there's always something that breaks the defenses. So one of the possible mechanisms is aging. And you can see that the mutant grows in this case. But and here we use the real numbers, right? So these are not just examples, these are real parameters that were measured. The thing is that look how slowly the mutant grows here. A laboratory mouse lives at most 1,000 days. So by the time the lifetime is over, this mutant that was appeared at the very beginning kind of didn't make it very far, right? This is 100 cells. This is the equilibrium level of cells in the mouse. So that would not result in anything dangerous. So that maybe there's something else that is going on with the mutants. And this is the second mechanism by which we think the barrier can be broken in the hematopoietic system. This is the effect of self-induced inflammation. So it has been noted that some of the mutants that rise in the bone marrow have the property of creating their own source of inflammation. You all know that inflammation is bad somehow, right? So inflammation plays a high role in cancer in many different ways. But in this particular context, mutants create inflammation in such a way that they themselves are not affected by it, but other cells are suppressed. So it's a very clever mechanisms of those mutants. And this is the reason we see those mutants, right? Because they came up with this mechanism of how they can rise. They create inflammation that makes everybody else less fit. So it rises their own fitness. In evolutionary theory, it's called niche construction. They kind of create the microenvironment around themselves that makes them more advantageous than they were. If we put that in, you can see that the mutants can rise in less than 1,000 days in the mouse. So this is a realistic scenario of how a mutant with a reasonable advantage of 2% can rise in the mouse in a reasonable time frame. So I think that's it. And I don't want to start a new topic, although I have a whole other section prepared. I think I would like to take questions right now, if you have any.
Can I start just by thanking you about how cool the combination between the biology and math is? These two lectures have been really, really enjoyable. Thank you so much.
Oh, thank you. I'm
Um,
so glad to hear this. This is one of the goals of these talks, to show you how exciting this area is.
Yeah, it is really, really exciting. I just have one quick question. So going back to the hierarchy, where you like introduced the system at the beginning, or maybe like, yeah, just,
Here?
yeah. So the question, my question is, it seems like gamma 1 and gamma 2 do not sum up to 1 in this model, although I would assume they would.
Do they not sum up to 1?
Yeah.
So why do you say they don't? We assume that they do.
They do? Yeah. I mean, I would have assumed that you would write them as 1 minus gamma.
Oh,
Like
yes,
gamma
yes.
and 1 minus gamma instead of gamma 1 and gamma 2.
Yeah,
Like,
I think you could do that.
yeah. It's just, I thought this will be one more
You're absolutely right.
parameter.
Yeah.
Yeah.
I didn't have to write it like this.
Yeah. Okay.
They sum up to 1. Yes. Thank you.
Yeah. Thank you.
Thank you. More questions? Okay. If not, thank you for your attention. They were long lectures, and I hope I convinced you that this is an interesting topic. Thank you very much. Bye.